Good evening, ladies and gentlemen. On behalf of the Faculty of Science, I would like to welcome you all to this inaugural lecture of Professor Alexander Valentine, Professor in the Department of Botany and Zoology. I would like to extend a special word of welcome to Professor Eugene Kluter, our Vice Rector, Research, Innovation and Postgraduate Studies, as well as Alex's wife, Tanya, and their two sons, Cade and Luke. Warm welcome to you. The program this evening will be as follows. I will introduce Professor Valentine briefly. He will then deliver his lecture, and Professor Konrad Mathieu, Head of the Department of Botany and Zoology, will propose a vote of thanks. Professor Valentine obtained his PhD in plant physiology from the University College Dublin in 1997. In that same year, he was appointed as a lecturer in the then Department of Botany at Stellenbosch University. He left the university in 2004 to spend the next 6 years at the Cape Peninsula University of Technology and the Nobel Research Foundation in the USA. Fortunately, in 2010, he was appointed as an associate professor in the Department of Botany and Zoology at Stellenbosch University, and he was promoted to professor in January this year. Professor Valentine's research focuses on mineral nutrition of plant-soil microbe interactions, specifically the adaptations of legume nodules to metabolize nitrogen during phosphate deficiency. He has published numerous papers in this field and serves on the editorial boards of numerous scientific journals, including BMC Plant Biology, PLOS ONE, and Scientific Reports. In 2017, he was invited to edit a special issue of Frontiers in Plant Science, one of the top plant science journals, on the topic of phosphate deficiency in plants. Alex's expertise has also allowed him to establish research partnerships with industries in the agricultural, mineral, energy and biotourism sectors, as well as international academic institutions. Currently, he is appointed as a visiting professor at the University of Temuco in Chile. To date, 10 masters and 11 PhD students have graduated under his supervision, and he is also well known as an enthusiastic and engaging lecturer. Currently, he is leading an initiative to develop an interactive e-book, which will also be available in Afrikaans. Professor Valentine has established and coordinates the outreach activities of the Department of Botany and Zoology, including the Bafunda Ilanga project, which provides support to resource-poor schools in the Western Cape, and also acts as a plant biology curriculum advisor to the Western Cape South Metropole schools. On a personal note, Alex has numerous hobbies and interests, including reading poetry and political science, volunteering, caving, and learning foreign languages such as Spanish and Mandarin. Ladies and gentlemen, please join me in welcoming Professor Alexander Valentine.
Attach the second one.
So is it on?
Yeah, it is on. And then this one. Attach it right here. And if you just put in your other pocket. Okay.
And when turn the volume up?
Okay. Yeah.
Thank you very much for the welcoming me and for presenting me as a professor at the university. I really appreciate that from the side of the university, my department and my colleagues. I would like to talk about the work that we've been doing in our research group. My topic of research is on phosphate deficiency in legume nodules, as Professor Valentine said, and I'd like to give you a short talk, hopefully a short talk, about this work. Now, my work essentially focuses on root biology. I'm a root biologist, and I've tried to make this presentation really not simplistic, but at least in a way that it's accessible for everyone. So for my postgrad students and my research colleagues, I apologize, I'm going to shrink the work that we've done together down to a single slide, but I would like to make it accessible for everyone. So if you look at roots, essentially, there are these underground structures that access nutrients and water for plants. And our focus of the work is on that part of the root system, the rhizosphere, which is the intimate connection between the root system and the soil, and that section is called the rhizosphere. So that is what my group and I have been working on for the past several years. So the talk will be focused on this section of the work. And as you can see, roots are engaged with loads of microbes below ground, which is why I have such good friends in microbiology with Alf Botha. So I don't think a lot of my research would have been as pleasant if I hadn't collaborated with people in microbiology. So how did I start in root biology? Well, I mean, I'm very glad that my honours supervisor, Professor Lincoln Wright, is here from UWC. I started off with mineral nutrition, working with Lincoln Wright, and I moved on to working on mycorrhizae with Bruce Osborne and Derek Mitchell at the University of College Dublin. I did my master's and PhD with them on mycorrhizae, still remaining below ground. I hadn't seen Bruce in probably 20 years, and I saw him in January again in Chile when we presented the same course for PhD students. In order to understand the changes in plant biology, especially the omics of evolution, I took a research fellowship for 2.5 years at the Nobel Foundation to work on Medicago legume nodules. So I started to switch from avascular mycorrhizae to Medicago in order to understand the Medicago nodule system even further. I then later went to China, to Shanghai, and I worked at the Plant Stress Center in order to learn more about proteomics in legume nodules. But you know, the thing about your first training always comes back. And with the Chileans, I started working again on the vascular mycorrhizae, and this was a trip we took through the Atacama Desert on bicycles to collect some vascular mycorrhizal plants. We've actually published a paper recently on that field work. So that is how I ended up as a root biologist, and I'll probably remain one until they drag me out of here. But let's have a look at what phosphorus is. Now, the point of the talk is to look at phosphorus and how phosphorus interacts with legume nodules, and how it enables legume nodules to fix nitrogen in nutrient-poor soils. So phosphorus is a soil mineral which is essential for plant growth, along with a complete suite of other nutrients. So if you look at soil, it's really a reservoir for a lot of mineral nutrients. And plants access all these mineral nutrients for a variety of reasons, but phosphorus is one of interest for us and for our group. So I'd like to focus this specifically on phosphorus. So what is phosphorus? Right? Phosphorus is a highly reactive chemical, and because it's so reactive, it's rarely found as a single element in nature. It's always bound to something like oxygen, or it's another compound. And because phosphorus is always bound to something, it's never biologically accessible for plants. It's always difficult for plants to access phosphorus. So normally the form in which plants access phosphorus is called phosphate. So I'll be referring to phosphorus in this talk as phosphate, because that's the form of phosphorus that plants acquire. Some interesting aspects about phosphorus is that phosphorus was discovered in 1669 by a German scientist, Professor Brand, and he isolated phosphorus from urine. He used something like 60 buckets of urine because he was trying to isolate gold, right? And accidentally isolated phosphorus from these 60 buckets of urine. And he called this new element cold fire because it glowed in the dark, and the name of the element comes from Phosphorus, which means bringer of light. Right? So some text also refers to phosphorus as the devil's element because of its eerie glow in the dark, its tendency to burst into flame, and because it was at the time the 13th element known. Right? Phosphorus in the form of white phosphorus is able to self-ignite at 30 degrees Celsius, and it's used in weapons and fireworks, and it's always stored underwater. Red phosphorus, however, is a lot more stable, and if you're a smoker, you obviously use it a lot because it's used as the striking surface of matchboxes, right? And that is when white phosphorus is slightly, when the red phosphorus, by force of friction, is slightly transformed into white phosphorus, and that provides the ignition to light the matchhead. But on a global scale, phosphorus is also worrying, especially for agriculture. And the reason is that the Earth may be approaching what we call peak phosphorus. And it's problematic because it doesn't mean we're going to run out of phosphorus, but it means that in 4 to 5 decades, the mining of phosphorus, where we currently mine phosphorus for fertilizer, is going to become unsustainable. And that is worrying. It means that we need to understand our plants cope with low phosphate conditions, because we are going to face a future where fertilizer phosphorus is going to become unaffordable for agriculture. And the reason why we are in that dire position is because phosphorus has an extremely slow geochemical cycle. It's one of the slowest geochemical cycles on the planet. It's because it's mostly terrestrial. All the other geochemical cycles have an atmospheric component, nitrogen, carbon, and water, but phosphorus doesn't. Phosphorus is made available when ancient marine sediments, because the marine sediments are made up of phosphorus deposits, and those ancient marine sediments are then made available either through slow geological events when water retreats, or when there are uplifting events where these sediments are exposed to the surface and we are mining it as we do today. And there are few places where we can actually get phosphorus from. I mean, the known phosphorus mines on the planet come from USA, Morocco, China, and a few in South Africa. And those are the places we currently mine for phosphorus. So with that in mind, we need to understand what is important for phosphorus in terms of life, right? Why is it important? It plays a major or an essential role in several areas. Phosphorus is part of genetic materials such as DNA and RNA. It's part of structural material, the phospholipid cell membranes, and it's part of energy and transfer reactions, part of ATP and protein modification. So it's essential for life. Now you may wonder why on earth we want to link, or we have been linking phosphorus with nodules. Well, it's essential for nodules because it enables nodules to grow, it enables nodules to function, and nodules are part of plants which belong to the group of legumes. Right? So what are these legume nodules? You've all probably encountered these legume nodules. If you've ever plucked up a bean plant or pea plant, those little pink nodes on the roots, those are legume nodules. So they belong to the family Fabaceae, the legume family, and these nodules are formed when bacteria from the soil form an association, a symbiotic association with the roots, so they live inside these nodules. And the bacteria provide the plant with the capacity to use atmospheric nitrogen. So as you're sitting there breathing in something like 78% nitrogen gas, it is an unusable source of nitrogen. But these organisms have the ability to convert that into gas, into a usable form of ammonium mineral for plant growth. Right? So that is why this symbiosis is so beneficial for plant growth. So if we want to look at a closer look at the nodules, we should use microscopy. And for this, we can open up the nodule. First of all, you'll see the nodule is pink. It's quite interesting why it's pink. It's pink because it contains a protein called legumoglobin. Right? You have hemoglobin coursing in your blood, so when it binds to oxygen, it is red. In the same way, the plant synthesizes legume hemoglobin to enable the bacteria to function normally, because the bacterial enzymes, especially nitrogenase, require a low oxygen environment. And that's why these nodules appear pink, something we share in common with in terms of our blood pigment. So to explain how that process works. I'd like to reduce the nodule down to this structure, so I'd like to show you just in a brief slide what it looks like and how it works. So within the big nodule, I'm going to represent one infected cell which contains the nitrogen-fixing bacteria. When these bacteria are inside the cell, they are slightly modified and they're known as bacteroids. So within the bacteroids, we've got the enzyme nitrogenase, which will convert this gas you're breathing in into ammonia, and thereafter ammonium as a mineral. But nitrogenase is sensitive to oxygen. So the plant synthesizes legume hemoglobin to bind the oxygen and keep it away from the reaction center of the bacteroids. Right? So that explains why they are pink. So how do nodules fix nitrogen during phosphate limitation? This is a question that we were wondering about for a long time, because if you look at the fynbos, you have all these wonderful legumes like rooibos, keerboom, and lots of beautiful legume trees and shrubs, and they fix nitrogen, but the question is, how do they manage to fix it in this phosphate poor environment? In order to study this problem, we looked at it in several ways. We started looking at it using model legumes. The beauty of model legumes is that their genomes have been sequenced and there are loads of resources available for research. So it's a nice treasure trove of information. But the problem often is that it's not applicable to what we have in our local environment. So we've also started looking at rooibos plants and the keerboom, Virgilia. The reason why we decided to do this work several-fold. First of all, we wanted to understand on a fundamental basis how these things work, some sort of blue sky research, which is important in society, but also the application thereof. If we can understand how these plants are able to fix nitrogen and metabolize nitrogen in a very nutrient-poor environment, these findings should and could have applications for agriculture. If we are facing a low phosphorus future, where fertilizer applications are going to become scarce and expensive, we need to have crop plants that are ready to use phosphorus in a nutrient-poor environment, to utilize phosphorus more efficiently. So we've been approaching this problem using legume nodules and using fynbos nodules, fynbos legumes, right? So how do they adapt to phosphate deficiency? Now this has been a long program that we've been involved in. So we started off looking at symbiotic adaptations. Does the legume root system engage in frivolous symbiotic associations in the soil in order to help the nodule obtain extra phosphate? Does the nodule change its anatomy in some ways to adapt to acquiring phosphate? And of course, something that I'm more interested in than those two, but those two are essential to investigate, is the functional adaptations. I was trained as a physiologist, so it will always come down to how do these things function. So I'd like to give you an overview of what we've been doing in these various fields. So let's start with the symbiotic adaptations. Now, for symbiotic adaptations, we can start with mycorrhizal fungi. We've also been looking at free-living microbes, and I'll take each one at a time to explain to you how they work. Now, with mycorrhizal fungi, you've all probably encountered fungi at some point in your lives. If you can think of the moldy stuff that grow on your bread, those are fungi. And these fungi can also come in a different form, same kingdom of organisms, where they can form a symbiotic association with the root system. So they become friendly with the root system, such as that, and they extend the surface area of the root system in order to allow the root system to access phosphate in far-flung areas of the soil. Right? It's not as simple as that. I mean, they have a complicated courtship of chemical exchange to allow the symbiosis to work. But in a nutshell, that's essentially what it is. And when you look at the mycorrhizal root interface, I think this was a photo from one of my PhD students, Karen Klutter. So this picture shows the mycorrhizal fungi colonizing the inside of the root system and using that interface to exchange nutrients with the host plant. So we have phosphate in far-flung areas of the soil, difficult to obtain via the normal root transport. So the mycorrhizal fungi with their hyphal extensions can reach phosphate where the host root can't, and they can acquire it for uptake. So if we now magnify a small part of this root system, I'd like to show you how this uptake actually works. So here we have the extended mycorrhizal hyphae, the root, and a nodule. So phosphate gets taken up, it gets exchanged for something like sugars, because in this symbiosis, nothing is free. So the host has to pay the fungus in some sort of carbon currency. It's usually sugars like sucrose or glucose. And then the fungus passes on the phosphate to the mycorrhizal, to the host, and the phosphate is then taken up, translocated all the way along the transport conduits of the root, and eventually to the rest of the plant. Now, something interesting happens when the fungus accesses phosphate. The fungus accesses phosphate and makes it available for the entire plant. But when the nodule gets hold of the phosphate, the nodule normally hogs all the phosphate and does not share it with the rest of the host. So the nodule is quite specific in what it wants out of this host-plant relationship. And the nodule normally holds onto the phosphate because it simply just has a higher requirement for phosphate to allow its functioning to occur. Now, the other type of symbiosis, and this is work I did with Alf Boerter at microbiology, is to look at free living microbes that form a type of biofilm around the root system to enable them to take up nutrients and help the plant in nutrient poor conditions. So this is what these yeasts, we worked on yeasts with Alf, what these yeasts look like under the microscope. So if I magnify this, I'd like to show you the biofilm of the yeasts, and we found in one paper with Alf that the phosphate can be stored in the biofilm as a reservoir, and the host can actually access this in times of stress. And although we haven't looked at it in a model legume system, in the other legume systems that we know of, the nodule is quite greedy and hungry for phosphate, and would most likely hog the phosphate for its own utilization. So we've now looked at one aspect of what we've been doing so far. We looked at the symbiotic associations and how these plants use symbiotic associations to enable the legume nodule to obtain phosphate in extremely nutrient poor systems. We've looked at mycorrhizal fungi, we've looked at free living microbes. So these symbiotic partners enable the nodule then to access biounavailable phosphate. So for us the next question was, are they able to cope without them? What if the nodule grows with a plant in an environment where it can't access symbiotic partners? And that started the next part of our program. So we've looked at symbiotic associations. Our next set of work was to look at anatomical adaptations. Now, for anatomical adaptations, we've done some work with some colleagues at Itemba Labs, where we've looked at the nodule in its large growth space, looking at its infected and non-infected cells. And you can see quite nicely here that these darkened cells are the cells that contain the bacteroids, and the non-infected cells are the clearer ones. So this is just a scheme to show you what the nodule looks like in terms of its function, a single infected cell. I'll come back to this, so I'll just take out some of those things and like to focus on some of these. So this shows us that within the infected cell, the nodule has bacteroids inside them. The nodule also contains an entire set of functional organelles, and these organelles have made space to accommodate this bacteroid-containing cell. Let's quickly go across. Okay, so if we look at the anatomical adaptations, we can see here that in a low phosphate cell and a high phosphate nodule, we've got differences in the infected cells. For example, low phosphate nodules have got fewer infected cells, as you can see there at the top, but the ratio of infected cells to non-infected cells is actually higher in the low phosphate nodule. So it's a type of adaptation that they have, that they realize the phosphate environment is very harsh to accommodate bacterial nodules. So they change their ratio of infected to non-infected cells to still be able to fix nitrogen, right? So if we take one of these infected cells and we have a look at the bacteria inside them, this is work we also did with another PhD student, and we can see that the infected cells are filled with bacteria on the inside. Okay, so that's a single infected cell with loads of nitrogen-fixing bacteria. And given that the metabolic environment inside the nodule changes so drastically when the plant is now stressed for phosphate, we were wondering whether the bacteria would be able to survive inside these low phosphate nodules, because there are changes in the sugar components, the organic acids, the amino acids. And to our surprise, the bacteria were able to maintain their populations. They didn't change to more efficient or less efficient bacterial species. They remained as the same bacteria. And work that Darren has been doing in the lab was to investigate how these bacteria in the free-living form are able to cope with phosphate stress. And that work I won't be talking about this evening, but the results are quite promising. These bacteria are quite well adapted for a low phosphate environment. So in spite of this harsh phosphate environment, they still remain as the same nodulating bacteria that they've started out with. So the next question that we started looking at in the lab was then, can the nodule anatomy be altered perhaps in another way? So for this, once again, we look at the nodule. Here we used CT scanner technology, which our university has at the Central Analytical Facilities. I must just say that, you know, one of the pleasures of working here is that we've got really nice toys to play with. I mean, you have to pay for it, of course, but we've got very nice toys to play with. And we've got them housed under Central Analytical Facilities. It was such a great investment, so you can have anything analyzed if you have money to do it, of course, but it makes it so much simpler. It's well organized, well administered. And when I told my wife that we had a CT scanner, she was quite jealous, because she said at the time at the hospital they didn't have one, and here we had one to play with for research. But I mean, I'm grateful that the university has one. So using the micro CT scanner, one of my PhD students was able to look at the volume and the cell size and the nodule occupation of the intact nodules, grinding it down or splitting it open, right? And the technology is quite nice. So if you look at one nodule again, you can see the scanner over here as it passes through the nodule. Millions of photographs, and then we can use those photographs to compile the 3D version of the nodule. We can calculate the sizes of the cells, the volumes of the nodule, without damaging the nodule at all. And what we found with high phosphate nodules is that high phosphate nodules have larger infected cells by volume. They have higher density of bacteroids inside the infected cells. And that is in contrast to what we have with low phosphate nodules. Low phosphate nodules typically have smaller infected cells by volume and lower density of bacteroids inside these infected cells. But remember, on the first slide I showed you, they adapt by changing the ratio of infected cells relative to non-infected cells. So they're still able to fix nitrogen even in this extremely harsh environment. So, I mean, I know that nodules can alter the anatomy, the bacterial presence in order to cope with phosphate stress. And the next question, as a physiologist, we wanted to know, does this change the nodule function? Does this change in anatomy affect the nodule function? So we've looked at symbiotic adaptations, we've now looked at anatomical adaptations, and I hope I'm at least showing you, trying to convince you that these organisms are incredibly well adapted to grow in low nutrient ecosystems. They can grow very well, and they can use a wide array of adaptations. But now let's have a look at the functional adaptations. So once again, we take a single nodule, magnify it, and we can look at its function. So in order to look at its function, we started considering what is biological nitrogen fixation efficiency. You know, when the plant and bacteroid convert the atmospheric gas to into usable nitrogen like ammonia, it requires a huge amount of expense in terms of energy, carbon investment, and some plants might be able to do it with less of a carbon budget or less of an energy budget. So we started calculating the efficiency of nitrogen fixation by using different types of bacteria, different types of legume species, so we can get the perfect match of host and symbiotic partner. We've also looked at phosphate recycling as a functional adaptation. You can imagine when plants are stressed for phosphate, if there's absolutely no biologically available phosphate in the environment, they are then forced to recycle the available phosphate, to reuse the available phosphate internally in order to survive in a low nutrient environment. We've also looked at how nodules adapt their nitrogen metabolism. In other words, where do they get their nitrogen from? Do they suck it from the atmosphere, or do they switch to perform a function like a root and just taking it from the soil, right? Which also has energy cost implications. And lastly, because when you take up nitrogen and you convert it into an amino acid, you are heavily dependent on organic acid metabolism. So these were the main themes that we've been looking at as physiologists for the past 10 or 15 years. Let's look at BNF efficiency. So when the nodules take nitrogen from the atmosphere, the gas we're all breathing in at the moment, and they convert it via nitrogenase to ammonia, and then ammonium, that has a huge energy cost. When we looked at model legumes, these are now the Fasciolis vulgaris, the normal common bean or the sweet pea, we found that the efficiency of these model legumes are not as high as our own fynbos legumes. We actually found that our fynbos legumes can fix nitrogen more efficiently in terms of nodule mass. So you look at the moles of nitrogen that has been fixed per unit nodule mass, so they're a lot more efficient in terms of that. They're more efficient in terms of fixing nitrogen per unit infected cell. They're more efficient at fixing nitrogen per unit or per mole of phosphate and phosphorus, total phosphorus, and also per unit or per mole of ATP. So they, whichever way you express it, the local indigenous fynbos legumes are extremely efficient at fixing nitrogen, right? And we were wondering what makes that happen. How do they do that? What makes them tick? So that started a whole series of new experiments. And this is work that we did with some colleagues and friends at the Tembe Lab, Wojciech and Jolanta Pravilovic. And we used a method called PIXI, which is proton-induced X-ray emission, to visually determine the distribution of several elements. In this case, it's phosphate. So when we grew the nodules at high phosphate, comparing it to low phosphate, we saw that there wasn't a significant change in the distribution of phosphate, which is quite impressive because it means that these phosphate-stressed plants were able to allocate and move their phosphate or available phosphate around so they could still function, right? But that, for us, partially explained the increase in nitrogen fixation efficiency. When we looked at iron, the distribution of iron in these cells, under low phosphate, as you can see there, the iron is concentrated in the infected region of the low phosphate cell. And this was quite astounding because the iron is part of several important proteins which enable and underpin nitrogen fixation. Remember I told you about the pigment that is able to bind the oxygen, the leg hemoglobin. That contains a heme group, so the iron we think was allocated to that and also to the enzyme that fixes nitrogen nitrogenase, because that enzyme contains an iron protein, a molybdenum iron protein, and another iron protein. So iron was sequestered for these functions within the low phosphate nodule. The other aspect we looked at to understand the increase in nitrogen fixation efficiency was to look at phosphate recycling. Now, for recycling, we once again looked at a huge nodule, we magnified it, and phosphate in its organic forms can be recycled, can be broken down to normal mineral phosphate. right? So how do they do that? Once again, how are they able to get phosphate from all sorts of sources back to normal mineral phosphate? In the cell, there are loads of organic phosphate sources. Sugar phosphates, phosphate as phospholipids, in RNA, in nucleotides, and the plant has a wide array of enzymes that are able to break down the organic acid, the organic phosphate, and recycle it down to mineral phosphate. So they can break down a wide array of organic phosphate sources via acid phosphatases. They can even break down RNA via RNases, and we've also found that in some cases the nodules can exude these enzymes, excrete them, so that they can also recycle some of the phosphate on the outside of the nodule. So they're being quite efficient at using the maximum amount of available phosphate. Okay, as you can see there, also RNA. And of course the RNA is not just plant RNA on the outside, it's RNA from all sorts of organisms, but the nodule is very efficient at recycling this huge reservoir of organic phosphate. And when we compared, as you can see here, there's a set of internal phosphate recycling reactions and a set of external ones. We found that fynbos legumes seem to recycle a lot more internally their phosphate than externally. So they enhance the activities of recycling inside their legume nodules as opposed to outside, which makes sense because it's a larger source of organic phosphate inside the nodule compared to outside the nutrient-poor fynbos soils. So the other aspect of recycling was work that another PhD student of mine did. For this, we also used the analytical facilities at the university. We used NMR for this to find the phosphate compounds and identify them in terms of the concentrations when the plant is stressed for phosphate. So if we look at phosphate stress membrane remodeling, you can see here that the cell membranes, I mean, we all, as we're sitting here, we rely on cell membranes to keep our metabolism intact and inside. But these cell membranes are made up of phospholipid bilayers. And these phospholipid bilayers contain a phosphate head and a fatty acid tail. So when the plant is stressed for phosphate, when it has recycled all of its organic phosphate, it eventually turns in desperation almost to recycling its phospholipid membranes. And it can replace the phospholipid membranes with sulfolipids. Right? How this affects the functioning of the nodules, we are not certain yet. But under extreme phosphate stress, they can recycle this. You know, I always admire the microbiologists. They always have the first access to all these cool toys. This was first discovered in microbes, I think like probably 10 years ago before we did it in plants, because the technology becomes available first. And this is the first time it was shown in plant nodules. So it's always nice to have access to microbiology colleagues so you can see what's going on in their field. Now, the other way in which the plant's nodules can functionally adapt is via altering its nitrogen metabolism. Right? And when I say nitrogen metabolism, it's the whole sum of taking up nitrogen from the environment, whether it's from the atmosphere, from the soil, and converting that to amino acids, making proteins, and sometimes recycling it even. Right? So that's the whole sum, what I refer to. But when the plant is stressed for phosphate, it reduces its reliance on biological N fixation. Right? So it just stops using the nodules to fix nitrogen, and it starts taking up nitrogen from the soil, whether it's available or not. If it's sparsely available, it still maintains some N fixation, but it significantly reduces its reliance on N fixation when it's stressed for phosphate. And the reason it does this is because to fix one molecule of N2 requires huge energy expense in terms of ATP, and also the cost of maintaining the functioning nodule to keep the bacteroids alive requires loads of organic acids and sugars. So the plant reduces its reliance on biological N fixation. It also alters the export of that fixed nitrogen from the nodule into a different format. Right? So usually it would export it as amide amino acids like asparagine, but in the case of stress, it changes the export to ureides. It can also alter the pathways in which nitrogen is assimilated. And this scheme I showed you earlier on, just to show you that under stress, we have a change in the source of nitrogen, so it doesn't rely on nitrogen from the atmosphere. The nodule can actually take up nitrogen from soil. It uses mineral nitrogen. It can also engage in changes in the amino acid pools. So instead of synthesizing loads of amino acids for export, it changes the source of nitrogen export, and it starts to export forms of nitrogen like ureates, which is quite a dense form of organic nitrogen. Other pathways which they change are the ways in which they assimilate and degrade nitrogen. Sometimes plants can use an enzyme called glutamate dehydrogenase to convert ammonium to glutamate. But what then happens when the plant is stressed, the plant actually allows the enzyme to function in the opposite direction, where it breaks down the glutamate to alpha ketoglutarate and to ammonium. What it essentially means is that the plant is slowly eating its own protein and nitrogen sources to generate huge amounts of organic acids, so that those organic acids can serve as a fuel to keep the respiration cycle going. The other aspect of our work, which I probably found the most enjoyable, was organic acid metabolism, right? So like I said to you, the organic acid metabolism is quite closely linked to the metabolism of amino acids, because when you have a nitrogen group, you need to stick it to an organic acid skeleton or backbone to make an amino acid. So this is quite tightly regulated. So our next set of work was to see how on earth is the changes in amino acid synthesis or changes in amino acid metabolism, how on earth are they linked to organic acid metabolism? And to study this, we looked at the branch point in glycolysis, which is PEP. And I'm sorry for the public if you're not familiar with this, but in order to explain this, there just isn't another way. So I'm sorry. But I hope you can ask me afterwards for any of these things. But believe me, I tried the whole week to make this accessible, but I can't see another way. Anyway, so we have PEP, phosphoenol pyruvate, which is metabolized either via PEP carboxylase or pyruvate kinase, and this changes the type of organic acid that can get imported into the mitochondrion and the TCA cycle. So what we've been doing for quite a number of years and using quite a number of PhD students was to look at the metabolism of PEP via these various enzymes. So normally when PEP is metabolized under, say, well-fed phosphate-rich conditions, the plant can utilize these ATP and ADPs, and it can import PEP as pyruvate into the mitochondrion and the TCA cycle. But when it's stressed for phosphate, it switches gears and it uses this bypass, because that bypass does not require, as you can see here, ATP and ADP. So when it's stressed for phosphate, it switches to a different pathway. And this pathway then uses different substrate, and it synthesizes loads and loads of organic acids in the form of malate, right? And as you can see there, it uses bicarbonate. And one of the experiments I enjoyed the most was when we used radioactively labeled C14 as bicarbonate to trace the incorporation of that carbon into various organic acids and to follow them through the various cycles, right? Various pools in the plant. And I really enjoyed that work in the lab. And the benefit of this is that the plant can still, or inside the nodule, can still synthesize pyruvate, but via malate using a different reaction. And this is a typical phosphate stress bypass. But you know, another benefit of this bypass is that the plant can generate huge amounts of malate. I mean, if malate sounds like a foreign term to you, if you've eaten a sour green apple, that is malic acid that you've tasted, right? So I mean, we at least can agree on that. You've all encountered malate. So that malate can also be used to excrete itself into the environment, so that the malate can liberate some of the phosphate that's biologically unavailable to plants. The malate, of course, is also a fuel for the bacteroids inside the nodule. Let's get these things to move. Here we go. So during this work, two of my first PhD students, Michalis Leroux worked on MDH, Alicia Kleinert worked on pepcarboxylase, and we've discovered some new things about malate. For example, we found that there were new isoforms of the one enzyme that converts oxaloacetate to malate, MDH, and that was really nice, interesting work. We've also looked at the regulation of pepcarboxylase in a lot more depth. And if we look at the aspect of organic acid metabolism, the enzyme that's important there is pepcarboxylase. Now, pepcarboxylase is the enzyme. It's a protein that consists of several polypeptides, and they can assemble into these fully functional enzymes consisting of 4 identical units, which we call a homotetramer. Right? But before this enzyme can be made to function and resist inhibition by all sorts of other metabolites in the cell, it needs to be phosphorylated. So the phosphorylation is a way in which, there are many ways in which the enzymes can be controlled and regulated post-translationally, but we're interested in phosphorylation. So phosphorylation is a way that allows the enzyme to be less sensitive to inhibition by other metabolites. So in layman's term, it makes it function better, makes it work faster. And to study the phosphorylation of the enzyme, we had to get an antibody which was very specific to the phosphorylation site when it was phosphorylated. We wanted to see the level of phosphorylation. And this was frightfully expensive to produce antibodies in that way. We collaborated with a colleague in Canada, Bill Plaxton from the Queen's University Biochem Department, and he gave us, as a very generous gift, antibodies that were specific to the phosphorylated state of the phosphorylation site. And with this, we were able to quantify the phosphorylation status of pepcarboxylase under phosphate deficiency. And we found that it was a major control point for the enzyme to allow the enzyme to function. We looked at the gene expression of the enzyme as well, the protein, and we found that the expression didn't correlate with activity. And when we realized that it was phosphorylation and the level of phosphorylation at low phosphate, it was quite a nice moment in our group. So there we've got the important role of pepcarboxylase and the status of phosphorylation, which controls the enzyme in these nodules in a way that is more important than its expression in terms of gene expression. So, we've now looked at nodules adaptation to phosphate deficiency in terms of symbiotic work. I remember I showed you the mycorrhizal adaptations and also the yeast work with ALF. We looked at the anatomical adaptations and we've looked at the functional adaptations. And I hope that this work has convinced you at least that these organisms are extremely interesting to study. There's so many facets of its function and its anatomy that can keep one busy for a long time. So these adaptations enable nodules to fix nitrogen in extremely phosphate-poor soils like in the Fynbos. So we've established that, and we think that is a benefit in terms of fundamental knowledge. But also, an additional benefit is that the mechanisms that these plants use, these nodules use to recycle phosphate, to use bypass reactions, to engage in symbiotic relationships, these things should be used for agricultural crop production. They should be exploited to breed new crops or to generate new crops with novel genes or enzymes that we've discovered from the Fynbos model. So our future work is to uncover more regulatory aspects of pepcarboxylase. When I was in China, we worked on some of the Fynbos nodules in China, and we discovered a new phosphorylation site on the enzyme pepcarboxylase, which was very, very exciting. So we need to redo the work again to be absolutely sure, but the preliminary data is very promising. So future work will be focused on looking at that. We also want to expand the capacity of biological end fixation to non-legumes. I mean, if you imagine that only the bean family, the pea plants, they're able to fix nitrogen from the atmosphere, they're able to use this reservoir of untapped nitrogen. Other plants like maize and wheat and barley, they're grasses. They can't fix nitrogen in the same way. So they need to be heavily fertilized, right? So I'll show you in a short while our preliminary data where we were able to expand biological end fixation to non-legumes. And we'd also like to investigate further upstream adaptations during pea stress. So far we've been looking at the end point of glycolysis, organic acid metabolism, but we'd like to look at the starting point where sucrose first enters the cycle. So we've recently, when I say recently, it's the last week, just published a very nice paper in the journal The Plant Cell. And the paper was about conferring the capacity of end fixation to rice plants. Right? What we did, and this work we did in collaboration with colleagues at the University of Calcutta, and we used a yeast which was infected with an endobacteria, and the endobacteria is of course able to fix nitrogen inside the yeast, and the yeast was allowed to form a biofilm around the root of rice, and we measured the end fixation, and indeed air from the atmosphere was transferred to the host plant. It's a proof of concept that microbes with the capacity to fix nitrogen as endophytes in symbiosis with yeast can be used as an inoculum to allow non-legumes to access atmospheric nitrogen. So indeed, the endobacteria fixed the nitrogen and transferred the fixed nitrogen via the yeast to the host plant. So to end off, I'd like you to have a look at this picture. It's a picture of beautiful nodules in a root system. And next time when you uproot a legume plant, I want you to have a look at those pink structures and appreciate the common pigment that it shares with your blood pigment, okay? And also the exquisite adaptations, if it's a fynbos legume, that they have to survive in nutrient-poor soils. For my acknowledgements, I would like, there's a lot of people I'd like to thank. I can't read all of them, but I'd like to thank my family, my friends, my colleagues, the university that has provided me with so much over the past few years. All my collaborators who have made the work really fun and interesting. And I'd also like to thank all my students. I think I was very blessed to have really great students. We were not without our arguments and fights in the lab, but they were really great students. The master students, my PhD students, and these are my current students. I would like to just mention that for my PhD students, I lost a PhD student this year. Lida Marie Groenewald was one of our really good PhD students, and she died under very tragic circumstances, and I think her brother is here, Yaku. So I hope that you were able to see some of the nice work that Lida has been involved with in our lab, and you know how things really will not be the same without her. So, thank you.
It is my absolute pleasure to first say congratulations to the real professor. Welcome to the club. I think this evening is a celebration to celebrate all your achievements, what you've done, and thank you for taking us through this memory road for you and a huge new thing to me. I was not aware this is all you've been doing on plants. But I think thank you for definitely establishing one of the world-recognized research labs in this area, or where phosphorus is needed for nitrogen fixation, if I got that right. And I think one of the things that really stand out to me is the collaborative nature of your research. If you're not in China learning languages, you're in Chile teaching courses, or you're at microbiology, or you're at horticulture, or not much with us, but you are around, and we value that. Thank you very much. I think it will have the thing stands out. And I also think we are really envious as we see your nice group of students. You always care about them, and they feel that at undergraduate and postgraduate level. Alex is always the one who steps in and says, I'll do it. And we really value that, and thank you for that. And then, I think if I can also maybe mention in this this whole thing of that you're always there for staff when we actually need you. There's no fuss towards science or people. Make sure that you don't have many enemies, and it's something that we also see and we also value in you, that you are always a pleasant person to actually work with, and thank you for that. And I guess I have to end off by actually saying we look forward to see you reaching new heights, and may it force be with you. Also my task to invite you guys. Apparently you are challenged to see if you can taste the mailer in the Chardonnays that's served outside. There are some of the refreshments you can go downstairs to please join Alex and us to interact with him a bit. And thank you all for also coming tonight. I'm sure Alex is very happy to see you all here. So,